What role does epigenetics play in DUX for gene expression for FSHD? Imagine your genes as a vast library of instructions, with some books kept closed to prevent chaos. Epigenetics is like the librarian that decides which books stay shut and which are open for reading. In healthy muscle cells, the DUX4 gene is one of those books kept firmly closed through epigenetic mechanisms. It's located in a region called the D/Z for repeat array on chromosome 4. Normally, this area is tightly packed into a repressive form of chromatin, with chemical tags like DNA methylation acting as the librarian's tools to keep DUX4 silent. This prevents the production of the DUX4 protein, which can be toxic if expressed in muscle tissue. In Duchenne Muscular Dystrophy, or FSHD, this careful repression begins to unravel. In FSHD type 1, the number of D/Z for repeats on chromosome 4 shrinks. This contraction causes the chromatin to relax, making it less tightly packed and more accessible. The DNA methylation levels decrease, weakening the epigenetic silencing. In FSHD type 2, mutations in genes like FSHD1, which are responsible for maintaining the repressive chromatin state, impair their function. This also results in the relaxation of the D/Z region, allowing DUX4 to be transcribed when it normally would be silenced. Once the epigenetic barriers are broken down, the DUX4 gene can be turned on in muscle cells. But there's an extra step needed for DUX4 to produce its harmful effects. The gene has a specific polyadenylation signal in the distal D/Z unit, especially associated with the FRQA haplotype. This signal stabilizes the DUX4 messenger RNA, allowing it to persist and produce the DUX4 protein. When this protein is produced in muscle cells, it triggers a cascade of damaging effects. It inhibits muscle cell differentiation, increases oxidative stress, and activates genes normally only active in germline or immune cells. All these factors contribute to muscle weakness and degeneration characteristic of FSHD. Interestingly, the expression of DUX4 isn't just a one-way street. Normally, cells have quality control systems like nonsense-mediated decay that degrade faulty or excess DUX4 mRNA. But in FSHD, DUX4 protein can inhibit these decay pathways, creating a feedback loop. This means more DUX4 mRNA and protein accumulate, worsening the damage. Understanding this epigenetic msrugdslutpun has opened doors to potential therapies. Researchers are exploring ways to restore the epigenetic silencing or block DUX4 activity, aiming to slow or stop the muscle damage. In essence, epigenetics acts as the gatekeeper for the DUX4 gene. When the gatekeeper fails due to genetic changes or epigenetic weakening, DUX4 is inappropriately expressed, leading to the muscle problems seen in FSHD. This connection between gene regulation and disease highlights how crucial epigenetics is in understanding and potentially treating muscular dystrophies. Always remember, if you or someone you know is affected, consulting healthcare professionals is essential for personalized advice and management.